Welcome to Phenomenal Science, the journey of a researcher with Phenomics Australia, a podcast brought to you by one of Australia's top research infrastructure providers. Here at Phenomics Australia, we support researchers to discover the genetic drivers of human disease and enable high-impact healthcare outcomes in precision medicine. Through our nodes across Australia, we offer specialised infrastructure, research services and technical expertise dedicated to advancing our fundamental understanding of health and disease. I am your host, Bridget, and I am currently studying cell and molecular biology and science communication. I have always been fascinated with genetics and how our lifestyles and environment shape us into who we are today. The interactions between our genes and our environment are what we focus on at Phenomics Australia to develop precise disease modelling tailored to individuals rather than a one-size-fits-all model. It is important that we can communicate the discoveries and impact of research to those outside the science community and educate and inform society on what we do. Phenomics Australia supports and guides researchers to pursue their passions through many ways we will discover as we follow the journey of researchers who are a part of our networks and use our expertise to conduct groundbreaking research into precision medicine. I would like to acknowledge the traditional custodians of the land this podcast was recorded on, the Ngunnawal and Ngambri people. We pay our respects to their elders past and present and recognise their continuing connection to land, water and culture. We also acknowledge the ongoing divide in equity within medicine and science with our Indigenous communities. Welcome back to another episode of Phenomenal Science, the journey of a researcher with Phenomics Australia. Today I am joined by Dr Lachlan Jolly, head of the Neurobiology Research Group and a First 1000 Days Fellow at the University of Adelaide, and a user of our Functional Genomics South Australia Phenomics Australia node. Dr. Jolly is a leading expert in neurodevelopmental disorders, investigating the fundamental genetic and molecular processes that govern brain development. His research focuses on critical cellular mechanisms like the nonsense-mediated mRNA decay pathway and protein ubiquitination, with the overarching goal to translate fundamental scientific discoveries into targeted treatments. Dr. Jolly's work is crucial for enhancing our understanding of these complex conditions, improving diagnostic capabilities and outcomes, and ultimately paving the way for tailored therapeutic interventions. Thank you for joining us today, Dr. Jolly. So to jump straight into it, what first inspired you to go into science and to begin the research that you're doing now?
The inspiration for science probably stems from my childhood, where I just had a passion of needing to know how things work. At a young age meant pulling a lot of things apart and trying to put them back together, or asking that question why. During high school, I obviously was drawn to science subjects, the chemistry, physics, and the math. And so through my high schooling, I fell to those subjects. What I'd actually never done during science, though, was any biology. It wasn't until I discovered that what I thought my career path was going to be, which is through engineering and a life in doing computer software engineering, I realised quickly that didn't really interest me much at all. And so I went back to the drawing board after a little bit of a hiatus where I tried some other career choices. But I ended up going back. You know what? I remember at high school I really liked science. So I went back to university and I chose all the science subjects. And for the first time, I actually did some biology. It was a molecular biology course that really fascinated me, and just that life inside of cells, what it looked like, how complex it was, how organised and chaotic at the same time it could be. That really drew me into understanding the molecular biology of the cell, and so I have a passion for cell biology. From that moment on, I just followed my passions.
Yeah, I resonate with that in terms of changing what you think you really wanted. When I was in high school, I was like, definitely I want to do chemistry. I thought that's what I wanted to do. And then I got into university, I did 2 years of chemistry and was like, that's not for me. And then I remember going back to my high school one time and talking to the teachers that I had and them being like, why did you pick chemistry? You were always better than biology. We could have told you this and saved you 2 years of pain in university level chemistry.
Yeah. No, it's hard, to see the tree for the forest when you're young and exploring it. There's nothing wrong with trying to explore, different pathways. But to follow your passion, I think it sets you up in life because you end up doing something that you really like. Work no longer becomes work, it becomes joyous in a great job where I get to learn something new every day, which, if I can do that for my whole career, I think it's just sensational. So just trying to fill up my brain with all the knowledge as much as I can fill it up in science is that industry. There's just so much cool science and cool technologies out there to learn. It's just never ending. So
yeah, really harnessing that natural curiosity of a scientist. And looking at your journey, like you said that you started off in your engineering and then moved into the science that you're working now, was there any moment in terms of when you were like, yes, this is what I want to be doing with my life, this is where I want my career to go, that sort of like a light bulb moment in your research?
no, I actually dropped out of university completely after engineering. I was just like, yeah, maybe I'm just going to go. And I also like using my hands. So, I was always building things in the shed or fixing things. So when I met my future-to-be wife, when I first got together and I thought, I've done this and I've done that, she's like, you got to go back. You've got a great opportunity, a golden ticket here. Why don't you go back and try something that you like? It was when I went there back to university and I did the bread of sciences, the maths and biology. That was the hook, the biology, as I mentioned before, that just really blew my mind. And from that moment on, I just directed all of my studies and thoughts down that pathway, and I just loved it.
How did you get into the research that you're doing now? Like you work with neurodevelopmental disorders. How did you discover that as an interest field for yourself?
It stems from my PhD project. So late in my undergraduate, I was lucky enough to have a laboratory placement, as many undergraduate students do. And I joined a laboratory that was working on a very curious enzyme. It was called a deubiquitinating enzyme, known as USP9X. This was a very boutique and quite sort of different type of enzyme that not many people worked on. We discovered very early on that this particular gene is really highly active in the developing brain. And so when I learned about this, I was really intrigued in the ubiquitination pathway, but also then I started thinking, what's the best way to study this protein and kind of reveal its mysterious nature? And it was, well, if it's most active in the developing brain, maybe we should use that as a model system. And so I took on the challenge of trying to understand the function of this enzyme in the context of developing brain cells. These are neural stem cells and the brain cells that they produce. And that just sparked my second kind of love in terms of, a science career into understanding brain development. I ended up getting an opportunity to go over to a Max Planck Institute in Germany and house myself there for about 6 months. And this is a world-class laboratory studying neurogenesis, this really primordial brain developmental stage. And that just blew me away. I just learned so much there in that 6 months. Come back and I was like, I just want to study neurogenesis for the rest of my life. This process through its brain stem cells create nerve cells. This is how your brain is built. And so I just developed that skill set. I showed that enzyme I was working on was really critical for the way that these stem cells build the brain. And from there, I finished my PhD. And the journey for me then was to try and apply my passion for neuroscience with something that I thought was a little bit more meaningful in terms of how could the knowledge that we're generating actually help individuals. And so I joined the laboratory of a guy called Joseph Getz, who was a geneticist. So he was a person who was looking at patients that had issues with brain development, that these are people who would present with intellectual disability or an epilepsy, and try and understand why. And usually if these problems arise during childhood, it's more than likely that the problem is of a genetic origin. And Joseph, he was a superstar geneticist, he still is, discovering these brand new genes that when they weren't working properly were the cause of these intellectual disabilities. So we married up at that point. I got to follow my passion in understanding brain development and actually apply it to better understand these disorders of brain development that are actually impacting human children.
I feel like brain development and your neurodevelopmental disorders have such a big impact on people's lives. If we think about society, a lot of people are accepting neurodevelopmental disorders more and they're becoming more aware of them in society. And I find, at least in the university system, there are more learning adjustments in place to support people who have neurodevelopmental disorders or intellectual disabilities and disorders. But we still have huge gaps in diagnosis and how do we diagnose people with these conditions. For example, ADHD or autism is underdiagnosed in females, but we also are lacking in the ability to actually do these genetic testing, that's really important. We can have all these treatments and these different therapies to help people, but how do we actually get them diagnosed in the first place?
Yeah, so the diagnosis can mean different things. I think based on the clinical features of an individual, you can get a clinical diagnosis, of ADHD or autism or intellectual disability, and that's relatively simple. It's just a matter of a clinician doing the appropriate assessments, and having that diagnosis can give you a path forward. It gives you an understanding, gives you a community as well. So it can just be a clinical diagnosis. But I guess what we're looking at in terms of the genetics of these disorders is really trying to put an exact gene-based diagnosis onto some of these disorders. Now, some neurodevelopmental disorders are complex, right? It's not a single gene disruption that causes disorder, but maybe thousands of small changes to genes which on aggregate have the outcome of autism. We call that complex genetics, where there's multiple genes involved or multiple gene variants. On the other hand, a lot of really severe developmental disorders, like children that will never walk or have 20 seizures a day, they're often caused not by complex genetic outcomes, but by single gene changes. So a loss of function of one particular gene can have quite a severe impact. You can imagine for an epilepsy, which is a disorder where the nervous system becomes overexcited. A lot of these genetic causes of epilepsy are the changes in genes for which the excitability of the neuron depends on. And then you have a drug, right? And the different types of drugs also will dampen down excitability, but by targeting these same types of gene products. So by understanding what gene is the cause of the epilepsy, you can actually triage what types of drugs to use and what types of drugs not to use because of their mechanism of action. And that's where you get towards what we call a precision therapy, where ultimately, if we understand what the gene cause is, then on the other hand, we then have a precision therapy tailored specifically for individuals with a disorder caused by that defective gene. And that's our goal, I guess, as researchers in this field, is to really come up with these really highly tailored therapies based on a genetic diagnosis.
So in the lab, you can study the different gene variants that you think are associated with a condition or a disorder. You did a recent paper all about skin biopsies and turning skin into brain cells so that we're able to study genetic diseases and disorders without actually having to physically cut into someone's brain, because for a lot of these disorders, is the only diagnosis pathway a brain biopsy?
It depends. The first pass in terms of genetic testing is to get a sample of blood. Now, every cell in your body has the same genome, so you can assess what the DNA changes are from a blood sample, and that's relevant to every cell in your body. When you get this genetic test, if it's a whole genome test, it gives a diagnosis to around about half of individuals. But we still have another 50% of people where we suspect their disorder is caused by a genetic problem. And what we can do is actually study the products of the gene. So every genome of every cell encodes every gene, but not every gene is active in every cell. And like you mentioned, if the gene is only active in the brain, then that requires a brain biopsy.
Which is incredibly... Yeah, brain biopsies are incredibly invasive, and a lot of people wouldn't be able to get one. So you're then left with a very high percentage of the population with genetic disorders being undiagnosed after just a blood test.
That's right. And so we started to think about how we could get around that challenge, because like you mentioned, the brain biopsy, and this is similar, like if it was a kidney disease, kidney biopsy is challenging, or if it's a heart disease. So we started to wonder how we could overcome this challenge of getting products from the gene from tissues where the genes are not active. And that's where we began to think a little bit laterally and start to really take notice of some recent Nobel Prize winning technologies that enable you to do a couple of different things. One of them was to turn skin cells into brain cells. And we can do that in the laboratory dish. And that's using the Nobel Prize winning technology called cell reprogramming. So the idea there is if we can turn the patient's skin cell into a brain cell, then the brain genes become active, and then we can then look at the products.
Which in of itself is pretty magical. Just thinking, changing a skin cell to a brain cell, it's two completely different cells. Like for me, that is mind-blowing technology to be able to have that. And then so many more capabilities, but can other parts of the health world harness of this new technology and these new discoveries and then use it in their own field?
Yeah, that's what we're exploring at the moment. We can make a brain cell. We use the brain cell as our kind of proof of principle, and it works really well. And we're helping diagnose people around Australia with this technology. And yeah, additional cell types are on the horizon on our hit list.
That's super exciting. And then you said there was a second method to this?
Yeah. So another way we thought about it, what if there was a way that we could synthetically turn any gene on that we wanted within a skin cell? So rather than turning the skin cell into a brain cell and then using the identity of the brain cell to activate the genes, what if we could kind of shortcut the process and deliver what's called a synthetic transcription factor, but it's almost like a gene switch. And we worked out this technique, another Nobel Prize-winning technology called CRISPR gene editing, which is used to introduce different gene variants across the genome. We worked out that we could modify that and turn it into a turn-on switch for genes. And so we tried that, and it worked really well. So now what we can do is we're now turning on brain genes in skin cells. And we can turn them on and we can get the products and we can help provide diagnosis to these individuals which are otherwise stuck in a bit of a holding place because they're challenging to otherwise resolve in normal diagnostic settings.
And how many people do you think will be able to help these sorts of genetic disorders? Like I've heard of something called like a variant or uncertain significance are really, really big in terms of this sort of work where it's genetic disorders, genetic diseases. A huge percentage of all diseases are these types.
Diagnostic testing now for individuals with developmental disorders is first-tier healthcare. The number of genetic tests that are being undertaken in Australia and also worldwide is just growing and growing and growing. There are literally millions, hundreds of millions of genomes that are being now done in a clinical diagnostic setting. Around about 50% of people still don't have an answer after that test, and a large proportion of those would need a technology like this to help resolve whether that variant that they've discovered is actually the cause or is simply a benign, suspicious-looking but nonetheless benign variant.
So it's a huge field and a really, really important thing for health overall and healthcare across the world. So if what you're able to do is then available worldwide, it will help even more people. Because as you said before, you have so many people going undiagnosed because you're unable to actually access the genes that are causing their disease or their disorder.
Yeah, the problem of variants of uncertain significance is growing and it's going to be challenging to resolve. Look, our study is addressing one aspect of this. There are plenty of other studies that are also looking at how we can provide additional evidence to support or discredit a variant of uncertain significance involvement in diseases. And it's the expensive part of the process. So we're looking for more opportunities to make high throughput scalable solutions. And ours is still very much in the research realm, but we are rolling it out as a national program.
And definitely, even if the upfront cost is high, when you think about, I guess, just the moral worth of this work and being able to bring those diagnoses to hundreds of thousands of people makes probably the upfront cost now worth it in the long run.
Yeah, it does. It's difficult to put a price on what a diagnosis can mean for a family, not just the individual, it's the whole family unit and often extended family. And then if you have a child, how you can care for it best. So it gives opportunities for prognosis, diagnosis, access to care. So it's very difficult to put a price on how much a diagnosis does mean. Now, we need to make these accessible to everyone. So there needs to be an economical solution. Now, we're trying to work with that to reduce our costs and also come up with other ideas to look at these inaccessible gene products to help diagnosis. Some people take that variant of uncertain significance and they say, well, I've got a diagnosis, I'm happy, because it means a lot to them. But in actual fact, it could be something else. It might stop them from going to do additional testing, find additional variants. So getting it right is really important.
Just giving them that here is potentially a reason for why you're experiencing what you're experiencing can be very moving and just super validating for someone as well. Like validating, okay, at least I can put a name to it. At least there is a reason, or in some cases, like, I'm not crazy, it's not in my head, it's like something that is actually here and it's been proven.
Yeah, there's so many reasons. You captured a few of them quite nicely, so thank you. You certainly understand the problem. We work a lot in rare diseases where there's perhaps only a couple of hundred people worldwide that are known to have disorder caused by this particular genetic defect. We find in those instances it is so rewarding for the families to then come together as a community. It's when those families come together as a unit and you have someone, a mother with a 1-year-old talking to a mother of an 18-year-old and saying, what can I expect from my child? Well, the mother of the 18-year-old is the expert because they've just been through it. So knowing what works, what doesn't work within these rare community diseases is super important, and that's where the community is really brought together by a gene diagnosis.
Yeah,
at a very minimum. Still what I'm really passionate about, and as I mentioned early on in my career, that move that I made to be involved in human genetics, to really have that outcomes of a research that are really impacting families, these are real life stories, is now what drives me. I can come to work and work on a variant and try and solve the puzzle of it as best I can. But when we solve them and we can send a report back to the patient's clinician, and the clinician then reports to us when we've reclassified the variant from uncertain to now we are certain, that's just a highlight in our laboratory when we get those moments. You can have certain days where a clinician tells you, thank you so much for your work. We've been working on this family for 20 years without knowing whether this gene variant was responsible or not. We've now told the family, they're just super happy, and then we've got smiles on our dials back here in the lab because we've made that connection to a family and made a real difference.
Yeah, super motivating, super rewarding work, definitely. So you're saying how you do a lot of work on different variants, so you do the gene studying. How do you actually, what does that look like in terms of how you actually study genes?
Yeah, so in the first instance, we actually deal with the genetic pathology and clinicians, pathology laboratories and the clinicians. We're trying to educate these health services that if you find one of these variants, one of these genes that's not active, please contact us because we may be able to help. And then we either one of two things, we can turn them into a different cell type, a brain cell, to access those brain gene products, or we can turn the gene on or off. From there, we then analyse the gene products that we're interested in. And that can involve some pretty high-end sort of DNA or RNA sequencing to really work out how these gene products are affected. And this whole process, from the moment the skin sample comes into the lab to when we can start to analyse these gene products probably takes about 2 or 3 months. We then assess the results to see whether the variant has a deleterious impact on the gene products, and then we write a research report and send it back. So the turnaround time can be quick, it can be long, but we'll do our best to return that result and hopefully provide them enough evidence that the clinician can then say, well, this variant certainly does have a deleterious impact on the gene. And we're able to now tell you that that variant of uncertain significance is in fact a likely pathogenic variant or a pathogenic variant. It's the cause of your disorder. Yeah.
And then you can start designing therapeutic treatments from that point onwards.
Occasionally, yes, and that's, there are different ways to try and revert the deleterious impact on gene products, and there are lots of nice emerging therapeutics that target the RNA part of the gene product that are looking very promising. These are things that we explore in the laboratory as well, not just trying to diagnose, but then going that extra step to working out how we can potentially use that information to design a therapy that can maybe alleviate the symptoms of the defective gene product.
And how does Phenomics Australia support your work, or how do you use our services? How did you come to know us? We have obviously our functional Genomics South Australia node at the University of Adelaide, where you're currently working. How do you use our services? How did you find us? What do we do in the role of your work? How are we involved?
Well, I've been a long-time user of Functional Genomics South Australia. The guy who leads the node, Professor Simon Barry, I used to actually share my laboratory space with him.
Oh, wow.
So we go back a long way. I was working hand in hand with him in a way for probably a good 5, 6 years at the Women's and Children's Hospital. And that's where I really started to utilise some of his expertise in a thing called lentivirus. Lentivirus is a way in which you can manipulate the cells in the dish. And we use the lentivirus to enable us to turn the cells into neurons or deliver things to cells to turn the genes on. So in both techniques, we rely on this thing called a lentivirus to manipulate the cells. And this was one of Simon Barry's fields of expertise. He worked on it in the biotech industry and then brought the technology back to Australia. And it's been one of the cornerstones of Functional Genomics SA is that they offer generating these lentivirus products as a service. So originally we were making these products ourselves in the laboratory, but these Functional Genomics SA are just so good at it, and they do it at cost. It just made perfect sense for us to partner with them. So FGSA was a partner on our very large grant. We were rolling these technologies out on a national level where we're recruiting patients all around Australia. Functional Genomics SA partnered with us on the application and a part of the funding. So we just use them constantly. I get an invoice from them every couple of months, and it's always very nice to pay it because I know it's very cheap compared to what we could do otherwise to other commercial vendors.
So it makes it a lot more economically feasible, and you know they're reliable, they know what they're doing, so you continue to go back every time. Do you use any other Phenomics Australia nodes aside from FGSA?
In the past, we've used another one here in South Australia, the South Australian Genome Editing Facility. You know, they specialise in generating and using the CRISPR editing tools to help us generate models of some of these genetic disorders. There's a few genes which are favourites of ours, which we take a deep dive into to try and understand. And that might mean sort of introducing a patient-inspired gene variant into a mouse or into a stem cell model, and then using that model to try and work out the biology of the pathogenesis and study it and then try and reverse it. So we've used South Australian genome editing several occasions to generate these patient-inspired experimental models in the laboratory that we can use to take a deep dive into trying to understand how these genes actually do control the way that the brain works and develops.
So you're not only just working with the non-animal models and like cell lines, organoids, but you're also using animal models. How are they complementary to each other? Do you see it as highly advantageous to have both working parallel to each other, or do you prefer one to the other? What do you think?
The models these days, you know, we primarily start with the patient, with the affected individual. So we very much use the human as the model wherever we can. And we can do some quite remarkable things with the patient cells now. As I mentioned, we can make neurons in the dish from them, or we can study various other aspects, but there are limitations. So if you want to look at the physiology of these biological systems, then you do need a physiological model like a mouse. So they're very complementary. You can ask different questions of different models. So we use a combination of stem cell models, mouse models, and patient cell models in our laboratory.
Do you see animal models leaving the science community anytime soon?
I don't think so. I appreciate that there are alternatives and they should be explored wherever possible. But having that biological system, I think, is pretty critical for development of new therapies and drugs. So I think they still have a very strong place in research, particularly for those that are really looking to translate new therapeutics to solve some of the most critical human health issues. The cell-based models, whilst they're getting more and more sophisticated, we can actually do things like organoids now where you're growing these brain-like structures or kidney-like structures in the dish, or they are now really sophisticated and are taking some of that load of how to test high-throughput therapies and things like that. But I think eventually, ultimately, you want to see how the physiology and the whole body system. So whilst you can have a brain organoid in the dish, you don't know how your brain drug might impact other systems like the cardiac or behavioural. You don't know whether a brain organoid has a speech problem. Can do that in a mouse. I think the cell-based and models like that are really good at looking at reverting molecular and cellular phenotypes. But when you want to have a look at whether your drug or therapy or your gene is doing at a whole organism level do a role for these small animal models or even larger animal models, they're going to be essential for, at least in some fields, maybe not all.
Cell lines, are they faster to produce than animal models?
Look, cell lines are very well suited to doing high throughput screens because you can grow cells in the laboratory almost endlessly, and that enables you to do lots of assays very quickly. So it's certainly at the start of something where you're trying to discover maybe what drug out of 20,000 FDA-approved drugs might be suitable for your particular cell-based model of a disease. The cells are essential for that purpose. You just couldn't do that with small animal models. But once you've narrowed it down to a couple of what we call like candidates based on this particular cell model system, these couple of candidate drugs or molecules seem to work really well. So let's now then triage them into a more sophisticated model into the mouse. So I see them more as probably towards the end point where you're trying to maybe work out whether your drug is suitable for treating an organism as opposed to treating a cell-based phenotype.
And so Phenomics Australia is able to help you in both sides in terms of you were saying Sage helps you with the mouse models and animal models, and then FGSA is helping you with your cell lines. So the capability we're able to help you in both.
Yeah, no, it's fantastic to have these services here. We're not also just limited to those that are within South Australia. We use the stem cell reprogramming facility at MCRI as well to generate some of these stem cell models. just good to go through the catalogue of places, I guess, on the Phenomics Australia website to see all the capabilities across Australia, where if you have a problem or you want to learn a technique or get some resources, it's a great place to go and check out what might be available and what services in Australia can be supported.
Yeah, because you're never limited to just one node just because you might use FGSA or SAGE most often. There are always all our other nodes that are available for use if they work in terms of what you're doing. So you've used our services of our two nodes, SAGE and FGSA. We also have a voucher scheme which you have recently been announced as a recipient, and you were saying that you have accessed our voucher scheme previously. I believe you said that you have done it with a mouse model. How have you found that?
I think it's an excellent scheme. It's actually quite remarkable that it even exists. It's quite a nice process to walk through in terms of application. And it supports you. These things like making a mouse model are not cheap. There are a lot of costs, not just with making, but also supporting that mouse over long periods of time. So any support you can get is very muchly appreciated. So the voucher scheme has been great. We use it to make a humanised model of a neurodevelopmental disorder. So we changed the genetics of the mouse to replicate what we discovered in some patients and really enabled us to take a deep dive into how this particular gene controlled the way that the stem cells of the developing brain of the mouse were altered. And without Phenomics Australia's support, I'm not saying the project would not have gone ahead, but it was certainly enabled us to do additional things because of the cost savings involved with the initial generation. And having those options available can be inspiring in a way. You might be sitting there, well, we could make a mouse or we could do that, but if you've got Phenomics Australia saying, well, we'll support you to do this, it can be quite encouraging and direct you down a path because you know the options are there and you have support from the experts in the field.
Yeah, we strive to be able to make it as accessible as possible. How did you become aware of the voucher scheme?
That was a bit of word of mouth. Just with my already existing relationships with the directors of both of those nodes, Simon Barry at Functional Genomics SA and Paul Thomas, who was another active collaborator of ours at South Australian Genome Editing Facility. They were the ones that alerted us these opportunities were available at the time and could help us support our projects that we had planned or were planning to complete with both of those nodes. So yeah, word of mouth.
I know that it's also promoted on our social media. If you subscribe to our Phenomena newsletter, you'll get in your inbox. We'll let you know when the next round's out. If you're interested, you can sign up through our website.
Yeah, these are all things I probably need to do. I certainly would like to have a heads up when these voucher schemes are released, put it on my radar. No, I just appreciate the opportunity to come on and tell my story and maybe the take-home message will be for listeners out there that if you have family members that have got a variant of uncertain significance, just to let them know that's not the end of your diagnostic journey. Please contact your clinician or your genetic pathologist and ask what's the next step. What else can we do with this diagnosis? There are plenty of programs out there that are trying to resolve variants of uncertain significance from different angles. Ours is one approach. There's a lot of push to try and better understand what these variants are in terms of a diagnosis, whether they are benign or not. So please don't take a variant of uncertain significance as your diagnosis. Please encourage people what else can be done to get a definitive diagnosis, which then carries the valley, ends your odyssey.
You're clearly very, very passionate about this work and very passionate to be able to give those people the answers to their questions and give people diagnoses. Is there anything you are most proud of?
I think if you're passionate and like I am about research, you'll always find a way to be involved one way or another. Trying to climb and become a group leader was challenging for me as an early career researcher. You go through this early career and mid-career stage where there are so many challenges to navigate and try and stay true to your research passion. So I'm proud that I managed to kind of navigate that through thus far. As a grant-funded researcher, you're always only as good as your last grant. But I'm proud that over the 15 years that I've been a grant-funded researcher, and I'm still here and I'm still able to follow passions and I have the freedom of research. So that's my persistence, but also the support of those around me that have enabled that. My mentor, Joseph Getz, people here at the Robertson Research Institute and the University of Adelaide that have had some faith in me at times where that was really, really required. And I'm proud of being in an environment that is supportive, and that is both driven by those around me, but now also creating my own environment. I started my own group about 2-3 years ago, and I'm really proud that that team, the camaraderie and the help that they supporting each other and support. So I'd just like to keep that legacy going from those that supported me to supporting that next generation of researchers, particularly through those challenging stages of PhD and early career stages where it's really, really important.
And really using your passion and to motivate others and support others to stay in a field that you love and continue loving to go to work every single day, I think is a really amazing thing, because not a lot of people get that.
Yeah, it gives you the bump, certainly does. There are rewards along the way that give you that little endorphin shot that keep going, and you're right, they may not happen all the time, so we also look at the little things that are important to us.
You were referred to me for the podcast by Jason, the head of FGSA. Do you have a close relationship with him? Is there anything you'd like to say about FGSA?
Definitely thank you to FGSA, and Jason is remarkable. He produces all these lentiviruses en masse, but he's just so passionate about trying to perfect this technique. It's really quite cool and inspiring in a way. So, every time we order a virus, he'll come back with a smile on his face if he's made the highest titer, which is essentially how you can measure how well the lentiviral preparation went. He often comes up with a smile when he's done a good job. So he's quite passionate about his role as well, and I think that's driving how well FGSA can support researchers both locally and perhaps more nationally as well. So you've got a good one there, a good scientist. So massive thanks to Jason. And like I mentioned, we've been working with FGSA, making viruses with them for probably close to 10 years now. I'm not sure how long they've been on note at Phenomics, but it's been a collaboration and a friendship across the journey. And so, it's always a pleasure to see you. We're in the same building. He pops up quite often to say hello and interacting with our researchers as well. So he gives tailored care. He asks what we're after, asks the questions, gathers information. He's quite nice. We do feel that when Jason's making our virus, he's doing it with love.
You feel like you're well taken care of.
So he just, like I mentioned, he just sent me an invoice today, and on the invoice, the facility has now made its 1,000th lentiviral preparation, and it says on my invoice, 1,000th ever lentivirus made at FGSA, and it says in bracket free. So he gave us a discount on that one.
It's fabulous to hear that you're having such an amazing experience with our staff and with one of our nodes. It's really amazing to know that our staff, they really strive to give the best experience to researchers and help as best they can. On a final note, is there any advice you would give to future researchers looking for somewhere to start? Any last words of inspiration you would pass on to a future researcher?
Future researcher is just, follow your passion. I think, as I've mentioned at the start, that's probably a key ingredient. So if you're really passionate about something and you have the opportunity to research in that field or satisfy that passion, then that's going to hold you in good stead. Take opportunities with both hands when they appear in front of you and put your whole heart into it. Science can be a tough game, but it's a super rewarding game at the same time.
Well, thank you so much, Lachlan, for joining our podcast today. I think that wraps us up.
Okay, thanks a lot. Appreciate it. Had a good time.
Thank you for joining us.
No, love it. Thank you.
And thank you for tuning into another episode of Phenomenal Science, the journey of a researcher with Phenomics Australia. I would also like to thank the Australian National University College of the Public Awareness of Science for allowing us to use their podcast room. Without the podcast room, this podcast would not happen, and we would not have access to their state-of-the-art recording studio, which I am very, very grateful of. Thank you for tuning in, and we hope you listen to the next episode of Phenomenal Science, the journey of a researcher with Phenomics Australia.